As we have been doing, we focus on doing topics together as a team. So the following purposes actually the drug discovery development. The very first one is to save lives, especially this time, this year, as Sophie raised, everyone was shocked. When you see the report back in February or not March, if you see, maybe you see some video clip, you see people walking down the street, and some people fall down, and later on dead. Okay, that is really a shocking scene in China, mainland China, and later in Italy, and later in the United States. So we see that happen. So the scientists were talking to do, right? And we have been doing drug discovery. Of course, we focus on doing what we can provide. So the second purpose is to improve health and living standard. And the third one is even more important and profit. Without that motivation, no drug can be in the market to benefit the patient. Okay, so what we have done, we quickly assemble, call out to the PIs, assemble teams. Our strategy is follows me. The first one is to synthesize Remdesivir. As President Liang also mentioned, in late January, there was a successful case using Remdesivir treating patient in Seattle. So we also, as a motivation, we have that synthesized in our hands. So to assure the people in Taiwan that just in case we have something that we can use. Okay? And the second one is to repurpose approved drugs, so we can have readily available drugs once it's identified effective against COVID-19. And the third one is to discover a new drug, and there are two different kinds of approaches. That is chloroquine and novel GC line pathways inhibitors. And we have several PIs integrated as a team for these three approaches. Chiung-Tong and Wen synthesized Remdesivir, and EO also focus on doing repurposing drugs. And another even more comprehensive team shows EO and this during this 20 minutes, my team, Chiung-Tong and EO shows they will also come here to the podium to present what they have done for fighting COVID-19. And during this time, we do research. However, we also publish papers, especially for drug repurposing, because everybody knows we do not have patients doing any further clinical studies. So, quick to contribute to fighting COVID-19 is to ascend, to present what we have found to the community, so everybody can follow our findings and to do further study. So these are the 4 papers we have published so far and 1 patent file. And this 28 is this paper, since it's online published in late May. So far, in 6 months, it's already been cited for 28 times. Okay. And this paper cited for 9 times and 3 times. I believe this is the very first AI paper published from NHRI. So we are very grateful that 立法院副院长 Legislative Vice President Chiung-Tong came to NHRI to encourage our young teams, especially to chemistry teams, Chiung-Tong and Chen. At that time, he mentioned that the two teams is like two light beams from a lighthouse. Okay, so shining over Taiwan. I think that's very encouraging to, especially to our young investigators. Okay, so I will give my time to Jimmy to be next.
It's my honor to share experience on COVID-19 project. And first, I would like to share the synthesis of Remdesivir. The second part, I will share the experience restricted to the new drug treating for COVID-19. So the first one, I received a mission code in early February while I was on vacation. And then I started collecting literatures and patents and starting materials for synthesis. 3 days later, I came back to Taiwan and launched the synthesis activity. And within 2 weeks, we complete mini bird scale. And then, at that time, President Tsai can encourage us. Also, 5 days later, we complete the synthesis of Remdesivir grand scale. Also, we overcome a very challenging step in the late February. It is the recrystallization of the chiral intermediate. This is essential for the future scale synthesis of kilograms scale. Also, we finish the 3.5 gram scale by using the crystallization process. Okay, and the Taiwan pharmaceutical manufacturers contact NHRI and want to work on kilogram synthesis of Remdesivir to help fighting COVID-19 at that time. So, the next one is, this is the synthetic skin. This one, okay, the first step is what I say, the recrystallization step. Actually, 3 days I finished carbon 5 for more than 10 grams. I can achieve this carbon in hand, but we spent more than 20 days to find a method for recrystallization. Even though we follow the paper, the patent, we cannot overcome this problem. But finally, we find a lower temperature condition. We overcome this problem. Okay. So actually, you can see the synthetic procedure is around 6 steps. But at that time, in Taiwan, we don't have these 2 study material, so we have to synthesize by ourselves. And we thank Tsinghua University in Chen, a professor, and also Zhongli University, Taiyou professor, provide us necessary reagents so we can start our synthesis of these 2 compounds. And then, within 6 steps, we can synthesize the Remdesivir. So the total step is around 18 steps. So the status of the Remdesivir is okay. On May, the US FDA approved the emergency use authorization for Remdesivir to treat COVID-19, and this is the brand name of Remdesivir, Biotechnology. is the first US FDA approved treatment for COVID-19 last month. So I shift to the new drug treatment for COVID-19. The approach one is structure-based drug design. Okay, we focus on the 3CL-like protease inhibitor. So we follow by the GC376, and then do the structure-based modification of this compound, and we can get the compound 7 is competitive with the reference compound, even in enzymatic or cell activity assay. We can competitive with this. So this compound is for the further investigation. Also, approach 2, we use AI-aided drug design. We use the AI platform for identification of this 3CL inhibitor, and then design a panel of compounds for the design. So this is the picture that we finished the crystallization. So everyone is very exciting at that time, and then we thank President Liang Dengli and Vice President Shitu Huikang and Dr. Chen gave us a chance to finish this job, and it is really an unforgettable and meaningful mission to us. So thank all of the teams. Thank you very much.
Thanks for Dr. Chen giving the opportunity to show you that the AI approach for COVID-19. We are not experts in this field. We just start to learn how to apply the AI to the drug discovery. In NHRI, we have set up 2 major platforms for AI application. One is for target identification. We hope AI can help us to identify some target effect or some side effect. The other is using the AI to assist the small molecule drug design. It includes within the activity, compound activities, and also assist the link optimization. Recently, we have a new breakthrough that we can perform the AI system to the new drug design. This year, we have a published 2 papers. This year, we have a published 2 papers AI application. One is about the program of TNBC, triple-negative breast cancer, and PCR. And the other paper is about COVID-19. The principle of our AI learning system is that after the learning data input, the system will use many descriptors to do the compound screen. And we put into the DNA algorithm. The algorithm will help us to identify the correlation of its activities. And it will also separate the important descriptors and give them different weighting. Then we use the system to use the different weighting number to generate the model equation. The model equation will perform through the database screen, and we use the in vitro assay to evaluate the predict result. After the in vitro assay test, the data will feedback for the system, let the system do the evaluation. After the COVID-19 pandemic, our President Liang has started one project is that our team tried to apply the AI system to identify some repurposing drug which can fight the SARS-CoV-2 virus. So at the beginning, we started SARS-CoV learning database. This learning database includes a compound which will inhibit the coronavirus. So we include CCL protease inhibitors, HIV drugs, some medical products, COVID-19 reported drug, SARS reported drug, RNA polymerase drug, and the flu drug. All these data will collect as the learning database. After the learning database includes our AI system generate two models, and we use these two models to screen the FDA approved drug. It's about 2,600 drugs. And then we evaluate by the non-COVID IPV assay. And we in the first round, our system predicts 60 drugs. And by the assay test, we identify four drugs have potent activities. And we feedback this data to the AI system and let the AI evolution and do the second round screen. For the second round screen, we screen 20 drugs for test, and finally we identify 4 drugs. So totally, by AI approach and with the IPV assay evaluate, we totally identify 8 drugs by the AI result. After our paper published, we tried to track in the AO drugs. We found that 5 of them have shown the SARS-CoV-2 inhibition by different groups their experiment test. And 3 of them have go through for the clinical trial. So this can prove that our AI system combined with the IPV visualization can efficiently to help us to screen the compound which can find the SARS-CoV-2 virus. Okay. I want to thank this team, the NHRI team, and especially want to thank the Korean team that help we can finish this job. Thank you.
We actually taking my order for fighting the COVID-19 this year. Many do drug or compound that has been reported by as antiviral coronavirus from literary research or Repurposing Taiwan Health Insurance prescription drug, as well as the protein-based compound broad-spectrum coronavirus inhibitor, both reported in my lab. We screened around 250 all drugs, and from them, 16 of them were we identified 16 of them as inhibitory activity against both blind coronavirus FFTV and human flu coronavirus OC43. And 12 of them were reported to have anti-SARS-CoV-2 activity by other research groups. And 4 of them also have a clinical trial for combating COVID-19 in phases 2, 3, and 4. We screened 244 Taiwan prescription drugs and identified 6 of them have very nice inhibition against human coronavirus OC43. And the one subject who SARS-CoV-2 activity assay not just got verified, but also worked in combination with Remdesivir in a synergistic manner as assay for OC43 and SARS-CoV-2 with synergistic score greater than 10, calculated by synergy factor. And the following compound was first reported by my lab that they have anti-broad-spectrum coronavirus activity. They highly inhibit the SARS-CoV activity with best EC50 smaller than 5 nanomolar, assessed by cytotoxic effect. And they also murine model. And since the outbreak of COVID-19 emerged almost a year ago, we went ahead to complete the test on the other two human flu virus, OC43 and 229E, as well as the most important. As well as the most important, the SARS-CoV-2 activity assay. For the three xenophoric-based compounds tested here, they fortunately reduced the viral titer from 7 log of magnitude to 0 as assayed for OC43 and 229E. They also properly inhibited SARS-CoV-2 as assayed by immunofluorescence. Assay and with antibody against the new capsid protein and the threat formation assay. And in both assay, our compound, the base compound, TD233B, all have very good EC50 around 14 nanomolar. And in comparison with Remdesivir, the EC50 in the same test, they have the EC50 is around 650 nanomolar. So it's about 47-fold better than Remdesivir. And from our previous work, we also have demonstrated xenophoric-based compound directly target the coronavirus genomic RNA in contrast with nucleic acid protein, which enable them to significantly, profoundly block the viral replication and transmission. So, in addition, we also would like to explore the combination of xenophoric compound with other drugs that has different mechanisms of action, such as Remdesivir inhibit the RDRP and GC376 inhibit the 3C protease. And we would like to move our order to complete our in vivo efficacy test on SARS-CoV-2 immune model and other required preclinical work to move our drug development further. And I sincerely thanks for all the superiors for their support and all the collaborators working in the SARS-CoV-2 and MITV and all the people who contribute to this work. And from about work this year, we have 2 publications, we have 2 paper publications and 1 US publication.